Hey everyone! Have you ever wondered what kinds of animals are out there in the wild? How would you find them? What if you couldn't see them? Well, today we're going to talk about using environmental DNA to figure it out. Let's get an introduction so we're all on the same page. Every organism has DNA. It carries information about how the organism will live and function. And as that organism moves throughout the environment, it can leave behind little bits of DNA. It's called environmental DNA, or eDNA. It can be things like hair, fur, skin, urine, saliva, and blood. It could even be mucus, like from the snail. And even the most rare organisms leave behind these traces. All a researcher has to do is sample the soil, the water, or the sediments to find the eDNA. This technique has been used a lot to study organisms that live in the ocean, but it hasn't been used on land very much. The researchers in this study wanted to know if they could use eDNA to find evidence of a really cool organism on land, the golden mole. It's hard to find them because they burrow, and there are 4 different species that live on the west coast of southern Africa. De Winton's golden mole, which is possibly extinct, the Cape golden mole, Grant's golden mole, and Van Zyle's golden mole, which is endangered. So the researchers used eDNA to figure out which of the 4 species were in the area. So how exactly did they do this? Let's get some methods. The researchers collected 49 eDNA samples by taking soil samples from 6 sites where there was golden mole activity, which they discovered using a trained scent detection dog. They also sampled DNA from 2 golden moles they captured in a pitfall trap, and from 2 golden moles that were killed by people's pets. Back in the lab, they extracted and sequenced the DNA and eDNA samples that they had. The researchers focused on 3 sections of the DNA that had unique sequences for each species, like a barcode. Then they matched the eDNA with the DNA samples they collected and DNA samples from a database to figure out which golden mole species they came from. So what did they find? Well, the researchers found all 4 golden mole species. 2 of the species, Grant's and De Winton's, they found in all 6 of their sampling sites. De Winton's golden mole is critically endangered and was believed to be extinct, so this is really cool finding. They also found possible evidence of Van Zyle's golden mole, which is endangered. What does this mean? Let's discuss. Well, first of all, this research shows that eDNA techniques work on land just like they do in the ocean. Second, they can detect rare species. The results show that De Winton's golden mole is not extinct. These techniques could help conservation efforts, too. Knowing where golden moles live will help people protect those areas. Now, they just need to narrow down the specific areas to target for conservation and figure out how many golden moles are out there. What about the big picture? Conclusions. Environmental DNA is everywhere. We leave behind a little bit of us everywhere we go, and humans are especially bad. There's nothing we can do about our skin cells sloughing off, but we can follow the basic principle of leave no trace by disposing of trash properly, not approaching or feeding wildlife, and not disturbing environments by taking things. Take a picture instead. These actions can help conserve habitats and the wildlife that live in them. Hope you learned something today. Have a good one.
Hey everyone, we're a small nonprofit and all of our resources are free. So if you like what you see, please subscribe and share.